Before I get started, I just wanted to go over a few housekeeping items. You can follow along on Twitter using the hashtag #RAForum. You can chat with other meeting participants using the chat feature. You can ask questions of our participants today using the Q&A feature. Please visit the exhibit hall booth or video vault to see all of the 23-1. All right, let's get started. Today I am joined by Dr. Willie Prado, whose bio includes, but is definitely not limited to, Vice Provost of Affairs, Faculty Affairs, Professor of Public Health Sciences, Director of Prevention Science and Community Health, and Dean of the Graduate School at the University of Miami. Dr. Prado also serves as a Research America board member and will be one of our esteemed judges today. Dr. Prado will provide an overview of our competition.
Thank you, Caitlin. The Research America Flash Talks competition is similar in design to the 3MT 3-minute thesis, a scientific communication competition that challenges scientists at universities to present complex research topics and their significance in a concise way. For today's competition, we have early career scientists in academia and industry who have prepared a 3-minute synopsis of their health-related research project, which includes the significance and context for their research. The goal of this challenge is to summarize a complex research topic to be presented concisely to a general audience. That's it.
Thanks, Joel. Thanks, Willie. Now I will introduce our judges. You've met Dr. Prado. He will be joined by Dr. CEO of Tripia, current Research America board member. Also, we'll be welcoming Jocelyn Moore, who served as the Deputy Democratic Staff Director for the Senate Committee on Finance, the former Executive Vice President of Communication and Public Affairs for the National Football League, and currently a venture partner at Ozone X Ventures. Later on camera to announce the winners. If you have questions for one of our contestants, please be sure to drop it in the question and answer so we can get to it from our audience Q&A portion at the end while the judges are deliberating. Without further delay, we're going to hop right in. Our first contestant is Jamal Alkhader. He is a PhD candidate at Weill Cornell. The title of his talk is Trending Now, a Netflix Approach to Drug Repositioning. Whenever you're ready, Jamal.
Let's take a trip back to the 1990s. Imagine that you're a middle-aged male and that you've just been diagnosed with heart disease. Concerned, you ask your doctor what options there are for you, and he proceeds to tell you of a clinical trial going on he believes you'd be the perfect candidate for. So you go and speak to those doctors and they agree. They start you on their drug, sildenafil. Now, after a few weeks, you go back to speak to these doctors and they proceed to ask if you've noticed any side effects since taking the drug. Blushing, you mention one rather specific side effect, lasting for no longer than 4 hours, of course. Sildenafil is more commonly known as Viagra today. Viagra is the perfect example of drug repositioning. That is, being able to take a drug originally intended to treat one disease and using it to treat another. Viagra, originally intended to treat heart disease, is now known to treat, well, you know. But why should we care about drug repositioning? Currently, it costs about $2.6 billion and takes around 15 years to get a drug from the small molecule stage all the way to market. Throughout this timeline, there are many opportunities for a drug to fail, and we don't want to waste our time and resources testing our drugs on random diseases, hoping to find that needle in a haystack. This is where we come in. Through my work in the lab of Dr. Olivier Elemento at Weill Cornell Medicine, we have been able to come up with a computational method that can successfully predict what disease the drug is able to treat. How are we able to do this? I believe that our method could best be compared to America's favorite pastime, Netflix. Now, whenever you finish watching a television show on Netflix, as soon as you've completed your show, you will be taken to a screen similar to this one, though with a different profile picture, I hope. Here, the service will then suggest other shows you might be interested in based on characteristics of the one you just watched. Because you watched a show with a strong female lead, you might like Wonder Woman. Because you watched a show about nothing, you might also like Friends. So let's apply it. There's a common expression. If it looks like a duck, and swims like a duck, and quacks like a duck, it's a duck. But in the drug universe, if it's structurally similar to a drug that treats prostate cancer, and has the same side effects as a drug that treats prostate cancer, and has the same targets as a drug that treats prostate cancer, then your drug could probably also treat prostate cancer. Through our work, we have been able to prove that drugs that treat the same disease do in fact share these characteristics, and more. So overall, we can take our initial drug in question, see which other drugs it is highly similar to in a drug network, see what diseases these highly similar drugs treat, and be able to predict a disease for our initial drug. And our method works. We've been able to see great success with our predictions. But we don't just want to stop there. We want to take this one step further and be able to make predictions on drugs where little information is known about them to aid with drug discovery efforts. So the next time you hear that you're a great candidate for a clinical trial, don't be afraid to join. But please make sure to tell your doctor if you're experiencing side effects lasting for longer than 4 hours, no matter how pleasant they may be. Thank you.
Excellent overview. A question ahead of time. Talk about a recent example of a reposition.
I'm sorry, Caitlin, I think you might have cut out.
Can you hear me okay?
Yeah, now I can.
Okay. A recent example of a repositioned drug.
Oh, yeah, for sure. So, I mean, the obvious one would be Viagra, but I'd like to actually draw, if I could, our lab was lucky enough to get our paper accepted last month into PLOS Computational Biology. So I'd highly encourage all of you to check that out. With our method is actually, it has a cool name. It's called CATNIP, which stands for Creating a Translational Network for Indication Prediction. But one of the reasons why it's great that it's called CATNIP is that actually CATNIP itself was something that was repositioned. Back in the old days, it used to be actually used as a poultice, you know, for skin lesions and other things like that. But once better medicines came along, you know, doctors didn't need that anymore. So they decided, hey, veterinarians were like, hey, we can use this in our tools. So let's take advantage of that, which I think is like a very, like, something everybody knows about catnip. But in our paper, we actually, when we looked at our clusters of drugs, we found that a lot of antidepressant drugs would actually kind of be highly correlated with Parkinson's disease drugs. So without giving anything away, that was something pretty exciting that with the resources we have at Weill Cornell Medicine, we were able to convene with experts in the field in both Parkinson's and diabetes who were able to look at our method and be able to be like, oh, this could potentially work. So very exciting stuff.
Awesome. Well, thank you so much for sharing that. We're going to go ahead and move to our next contestant. Let me go ahead and advance this slide. Medina Colic, who is currently a PhD candidate at the University of Texas MD Anderson Cancer Center. Her flash talk is titled An Avenue for Therapeutic Opportunities in Cancer. Medina, whenever you're ready.
Understanding how genes interact with each other and how genes interact with drugs has the potential to explain why patients with the same tumor type respond differently to a given treatment. Until recently, exploring these interactions was limited only to simple model organisms like yeast, because there was no proper technology to facilitate such studies in more complex organisms. With CRISPR technology, now we can perform similar studies directly in human cells. CRISPR allows us to edit the genome of a cell. The genome, think of it as being an instruction manual of a cell. Studying how genes interact with drugs in cancer cells is enabled by combining CRISPR editing with drug treatments. The major gap in the field was a lack of a method that can identify and characterize these interactions. To address this gap, I developed a computational method called DrugZ. The purpose of this method is to identify gene mutations or gene changes that enhance or suppress the activity of drugs. For example, using our method, novel interactions between two gene complexes and drug that inhibits the PARP enzyme were discovered. PARP inhibitor is a targeted therapy primarily used for BRCA-mutated advanced ovarian or metastatic breast cancers. Mutations in one of these gene complexes, called ribonuclease H2 complex, increase the sensitivity to PARP inhibitor, while the mutations in the other complex, called shielding complex, create the resistance to PARP inhibitor treatments. The second component of my research is investigating gene-gene interactions using the advanced CRISPR technology, which allows us now to edit more than one gene at a time. Think of it as being able to edit multiple pages of instructional manual at the same time. The most interesting and widely studied gene-gene interactions are synthetic lethal interactions. And these are interactions in which the deletion of both genes leads to cell death, whereas a deletion of one of these genes does not. However, very few of these synthetic lethal interactions have been reproduced across multiple studies, and many of them appear to be highly context-specific. Hence, the major drawback is the lack of gold standards or reference synthetic lethal interactions, and a need for a generic and robust method that would identify these interactions. And that is what I'm currently working on. In the preliminary studies from our lab, we have shown that the paralogs, which are a class of homologous genes, are the rich source for identifying these reference synthetic lethal interactions. Successful completion of this work has the potential to revolutionize cancer therapeutics by providing an avenue for generating new therapeutic targets, improving drug efficacy, and repurposing existing drugs. Thank you.
Thank you so much. Okay, so your question while the judges are deliberating is, I'm just curious, what other applications might this platform be used for? It seems like there might be other scenarios in which this can be potentially a therapeutic opportunity.
Yes, definitely. As I said, also our method is publicly available and free to use and redistribute. Another case where we can use the DrugZ method is to compare the isogenic pairs of cell lines. And the isogenic pairs of cell lines are the actually very powerful tool for understanding gene functions. And just to kind of catch everyone up, the isogenic cell lines are the cell lines that differ by one single genetic modifications. It's like 2 instructional manuals that differ by one single page.
Great. Well, thank you so much for sharing your work. Now we're going to go ahead and move into our next talk, which is with Amelia Dunn, who is a postdoctoral researcher at the University of Pennsylvania. And the title of her talk is Testing Opioids for the Treatment of Cocaine Addiction. Amelia, whenever you're ready.
I don't think I need to go into too much background detail about what a tremendous public health issue addiction is in the United States right now, especially considering the fact that less than 15% of people who meet the criteria for substance use disorders are currently in treatment. So one really important way to improve these numbers is to increase access to the current available medication treatment strategies. Another really important avenue is to research the underlying mechanisms of drug addiction and use that to try and develop new treatment strategies and new medications. And so that's what I spent my PhD at Rockefeller University studying. So there's a wide range of drugs of abuse that are addictive, things ranging from nicotine and alcohol to heroin and cocaine. And what all these drugs have in common is that they trick our brain into wanting to continue to use them over and over and over again, right? Despite all the negative consequences that they cause. And they actually do this through a common mechanism, which is the release of this chemical dopamine in the brain. So dopamine you maybe have heard of as this kind of feel-good chemical, and it causes these feelings of pleasure and euphoria that we associate with drugs of abuse. When scientists were studying opioid drugs, they actually found an entire class of opioids called kappa opioids that do the exact opposite. So kappa opioids actually block the release of dopamine in the brain. So you can imagine that if we are blocking the feel-good chemical, kappa opioids on their own actually make people feel really, really terrible. But if somebody's using a drug of abuse already, like a heroin or cocaine, that causes this massive abnormal release of dopamine, our lab's hypothesis is that we could use a little bit of these kappa opioids to block that abnormal dopamine release and maybe balance the dopamine scales in the brain and block some of those positive effects of drugs of abuse. Specifically, we're interested in seeing if kappa opioids can block the positive effects of cocaine. While we do have medications that we can use right now to aid in the treatment of nicotine and alcohol and opioid addiction, there's not a single FDA-approved medication for the treatment of cocaine or any kind of psychostimulant use disorder. So to test out our hypothesis that kappa opioids can block the positive effects of cocaine in the lab, we used an experiment you can see on the bottom of the screen with a box with 2 very distinct sides. And through a series of injections, the mice learned to associate one side of the box with cocaine. And under normal circumstances, the mice vastly prefer to spend all of their time in that cocaine-associated box, which we interpret as them really liking the cocaine. It has a pleasurable effect. But when we do this exact same experiment and treat our mice with a little bit of this kappa opioid, we find that they no longer prefer the cocaine side of the box. And so we interpret this as meaning that we've used our kappa opioid to block that pleasurable liking feeling of cocaine. So there's a lot of work that still needs to be done on this project. This specific kappa opioid actually has some really nasty side effects in humans. So we're working on discovering novel kappa opioids that maybe one day can be used in the treatment of substance use disorders in humans. Thank you so much.
All right, thank you for that overview. And with just enough time to spare. Okay, so I think this is such an important topic, and opioids has come up during other portions of our forum for the past few days. How do you view your work in the context of public policy, and what do you, if you want to comment on kind of the stigma and around helping patients find treatments?
Yeah, I think that that's a really big issue in the treatment of addiction. So, you know, there's a lot of evidence out there that shows that medication-assisted therapies, especially for opioid addiction, are really, really beneficial. But people have a lot of trouble getting access to these therapies because of that stigma. And I think that scientists have a really important role to play in kind of teaching and explaining the science behind that to kind of reduce the fear and the stigma around some of these kind of drug replacing drug type of treatments.
Yeah, I agree. Well, it's such an important topic, and we're thankful that you joined us today to share a little bit about what you're doing to work on that. So thank you so much.
Thank you.
Our next contestant is Brian Cantor. He is a PhD candidate at Rutgers, New Jersey Medical School. And Brian's talk is titled Systemic Radiotherapy for Breast Cancer That Spreads to Bone. Brian, whenever you're ready.
Thank you, Caitlin. Radiation is a powerful, cost-effective tool for treating cancer. About half of all new cancer cases are treated with radiation. Often a machine aims a beam of radiation at the tumor site. This can be effective for eliminating the tumor in one spot, but what happens when the tumor spreads as cancer cells slip away undetected? Cancer is a systemic disease requiring a systemic treatment. My research examined radiation as a systemic treatment for breast cancer that spreads to bone. I treated mice with a salt solution containing radium, a radioactive element. My control mice received only a salt solution and no radiation. Radium acts like calcium and localizes to bone. Next, I inserted human breast cancer cells into the large bone of the mouse's lower leg. The human breast cancer cells were deposited specifically into the marrow cavity. I found that human breast cancer cells inserted into mice treated with radiation were more likely to experience damage to their DNA and cell death. How exactly, though, is radium causing these cancer cells to die? Radium is unstable, and while decaying, releases energy as alpha particles. Alpha particles can be stopped by a thin sheet of paper, yet when they are ingested, the consequences can be fatal. It was alpha particles emitted from radium in glow-in-the-dark paint that was responsible for the deaths of young women watch dial painters. Alpha particles transfer much of their energy in a short distance. When an alpha particle hits a cancer cell, it can pierce the DNA, causing an explosion of damage to the double helix. Unable to repair the damage, the cancer cell dies. But in my mice, some breast cancer cells were farther from the bone and could not be hit by an alpha particle. How did these cancer cells also experience damage to their DNA and cell death? Breast cancer cells struck by alpha particles communicate with their counterparts that are farther from the bone and radium. The biological changes caused by radiation can be signaled from one breast cancer cell to a nearby bystander breast cancer cell. Having the ability to induce biological changes in breast cancer cells without directly hitting them allows radium to create a toxic space in the bone. The toxic space can prevent breast cancer cells from spreading to the bone. This matters for breast cancer patients, with about 20% of them suffering from cancer cells spreading to another site. 70% of those patients have breast cancer cells localized to bone. Ultimately, systemic radiotherapy with radium could help extend the lives of these patients.
Thanks, Brian. So thank you for enlightening us on systemic radiation. My question for you is, for many patients, this may seem like a scary treatment option. So what are some risks and benefits of radiation that you want to share?
Yeah, thank you. I think it's an important question, Caitlin. So we are very aware of, as a scientific community, what radiation can do at very high doses. And I think where the complication comes is at lower doses. But in terms of for cancer treatment, as I said in my talk, radiation is used to treat over about 50% of all cases. And so I think it requires sort of a relationship of trust between the clinician interacting with the patient, and also with the research community working with the clinician and with patients and patient advocacy groups to communicate what is known about radiation interacting with humans, and what is not known, and what we as researchers are working on to fill in the gaps.
Great. Well, I think that's such an important point, and we appreciate your insights on that. So thank you very much. Next up, we have Catherine Messina, who is a postdoctoral research fellow at Genentech in San Francisco. Her flash talk is titled, "Could Synthetic Polymers End Cold Chain?" So over to you, Catherine.
$15 billion. That's how much the transportation of vital medicines under refrigeration, known as the cold chain, cost in 2018. Imagine a world where we don't need to keep these medicines refrigerated. Vaccines could be more easily transported to places that don't have established infrastructure. Diabetics wouldn't have to worry about whether or not their insulin was still effective while managing everything else in their life. We could save billions of dollars per year, lowering the cost of and increasing access to healthcare. My research aims to make this a reality using synthetic polymers. When you think of polymers, long molecules made of repeating units called monomers, you might think of plastics like polyethylene, fabrics like polyester, or naturally occurring polymers like our DNA and proteins. Man-made or synthetic polymers can actually be really useful for applications in human health, because in making them ourselves, we're able to carefully design them. My research uses synthetic polymers to stabilize important medicinal proteins like insulin. My research group designed and made polymers based on the naturally occurring sugar, trehalose, which is used by many organisms to survive harsh conditions. Through my research, I was able to show that insulin could still effectively lower blood glucose in mice, even after nearly boiling the protein for half an hour, but only when polymer was present. Through biochemical tests, I showed that trehalose polymer was able to prevent aggregation of the protein, which could otherwise lead to issues like inadequate dosing or immune response, which are potentially dangerous situations for diabetes patients. We also saw that even 60 times the amount of polymer typically administered to mice was non-toxic and did not cause immune reaction. I also showed that you could alter how long insulin remained in the blood of mice by whether or not you attached the polymer to the protein, enabling us to make stable, long or short-acting insulins, which are both needed by diabetics to carefully manage their blood glucose. We're continuing this research looking at other important medicines containing proteins, like antibodies and vaccines, and evaluating exactly how the polymer stabilizes proteins. From these preclinical studies, we hope to translate this into real clinical applications. With these innovative polymers, we imagine that one day we can lower the cost of, increase access to, and maintain the safety and efficacy of these important medicines. Thank you.
Great. Well, thank you so much, Catherine. I know I learned a lot just from that short segment. And I think this application seems like a great one. So I'm wondering if there are other applications of synthetic polymers used in healthcare, and if you wanted to expand on that a little bit. And are there any that patients may not think of that as readily as you might think of?
Yeah, there's lots of really interesting research going on with polymers and synthetic polymers in the realm of biological applications. One example that I mentioned is making extended release or like long-acting formulations, which enables us to decrease the number of injections that patients need, which is definitely better for them because no one wants to get an injection. There's also really interesting research with smart materials, which is something that I also worked on in my PhD, which uses polymers that respond to different stimuli. For example, heat or glucose concentration, you can make materials that change their size and release a drug in response to these different biological stimuli.
Great. Well, thank you so much for sharing your work. And we're going to go ahead and move into our next talk. So for our next talk, we have Jordan Cockfield, who is a PhD candidate at the University of Notre Dame. And the title of her talk is Mapping Cancer Cells. Jordan?
Maybe I'll go ahead and get started. So imagine for a moment that you just hopped in your car and you're heading to arguably the happiest place on earth, Disney World. Now you've driven to Disney numerous times before, but suddenly the road you normally take is blocked and you're forced to search for another path. Now, unfortunately, your cell phone is dead and you don't have a charger, so no Google, no Siri. And as you contemplate on which road to take, the reoccurring thought in your mind is, I can do this if only I had a map. Well, you aren't alone in this thought. In fact, numerous oncologists harbor the same sentiment as their patients return to them because their cancer got worse. What started out as a primary tumor in an organ like the lungs has now mobilized or metastasized to an organ like the brain. And since we know that metastasis is responsible for 90% of deaths caused by cancer, it is at that junction that oncologists rely heavily on accurate research to help guide them to and through the unknown roads that lead to killing mobilized cancer cells. My name is Jordan Cockfield, and I'm a basic cancer research scientist in training. But for a moment, you can think of me as a map maker. Now, in order to understand my map, you have to know this one thing. Mobilized cells are supposed to die. And although there are some mobilized cancer cells that have found ways to cheat death, there are other mobilized cancer cells that don't have this ability. So my focus is on understanding exactly how that second group of cancer cells are dying, so that with that knowledge could come insight into making that first group of cancer cells play by death's rules. But before we get there, we must first understand the exact processes that are involved that lead to the death of mobilized cancer cells. So if you take a look at my map, what you'll see is a layout of a cell death pathway in mobilized cancer cells that involves them overeating their mitochondria, which you all know is the powerhouse of the cell. The mitochondria are eaten in a specialized compartment that you could think of as the cell's stomach. And we believe that as the mitochondria are being eaten, damaging chemicals are being released, and they poke holes in the cell's stomach. And out of those holes flows a special group of protein that's spread across the entire cell. And it's perhaps that special group of proteins that are responsible for killing mobilized cancer cells. So taken together, the significance of our work is truly having the possibility to guide oncologists through a cell death pathway that leads to killing mobilized cancer cells. So if you allow me, I'd like to join you back down your road to Disney, where I would leave you with this insight that I would give any oncologist, and that is, in order to know where you're going, you not only need to know where you've been, but you also need a map. Thank you.
Wow, excellent job. Thank you so much. And I just have to say that I don't know what I would do without Google Maps. So happy that you that you're working on that for cancer cells. So you've clearly demonstrated interest in science communication, and I know you wanted to share sort of what inspired this interest and what are your plans for when you finish your PhD?
Yes, so I think that's a good question, because I'm of the school of thought that I think the best thing that you can do in life is to find joy in the work that you're doing. And I think one of the ways that you can begin discerning what work you would have enjoyment in is by asking, what would you do if you weren't getting paid for it? Now, although I want to get paid, I think I answer that question by saying I would read scientific articles, I would conduct experiments, and I would travel all around the world discussing my research that I've done with collaborators, specifically on the understanding and advancement in therapeutic resistance in cancer cells or cancer patients. I think that, or I know that therapeutic resistance is a huge problem in the clinic as to date, and there's just incredible opportunities for more and more research and understanding to be developed that leads to a greater strategies or solutions that can be brought about in this arena. So if I had the opportunity to do something like that with my life, I would definitely find joy. And then I think the added bonus is if that work ended up helping somebody, that just enhances the joy for myself. So I think immediately after the PhD, I want to go into a postdoc program where I would be trained as a translational cancer researcher so that I could serve as that nexus between the basic cancer research that we learn at the bench and between the practical application for the patients. I think that's what I would like to do after my degree.
Well, that is just really fantastic. And I hope that you will continue to explore applications for your work and in order to help patients, because I think there's a lot of potential here for being that conduit. So thank you so much for sharing with us your research today. Okay, so for our next talk, we have Tyler Cambis. He is a PhD candidate at the University of Nebraska Medical Center, and the title of his talk is Using the Wrong Fuel for the Right Job. Tyler, whenever you're ready, please begin.
The energy crisis in America is a hot topic. At the center of this debate is the question: how do we choose the most efficient fuel source while being conscious of the health of our environment? My thesis is centered on finding the answer, but on a smaller scale and in a different setting. Instead of focusing on what fuel source is best for the health of the earth, my research focuses on what fuel source is the best for the health of the heart. 10% of the American population has diabetes, and thus are at an increased risk of developing heart disease, even if they use insulin. This problem occurs because diabetic hearts aren't able to use their normal fuel source, glucose, which burns quite cleanly, so to speak. They are forced to rely on an inefficient, dirtier fuel known as fatty acid. Relying on fatty acid as a primary fuel source results in increased fat deposition within the heart's tissue, which actually precedes diabetic heart disease. This has led many to believe that if we halt this fat deposition, then we might altogether be able to halt diabetic heart disease before it starts. And that is exactly the problem my thesis is focused on exploring, while taking the help of a very tiny molecule known as microRNA. You see, the microRNA that I work with protects heart function and prevents heart cell death. Interestingly, and for reasons that we do not know, levels of this microRNA are decreased in the diabetic heart. Thus, a potential solution to the problem of diabetic heart disease is presented. What if we took this microRNA and restored it to normal levels in the diabetic heart? I found that when we do just that, heart scarring is decreased, cardiac output is increased, and cardiac muscle contractility is restored, similar to if diabetes wasn't present at all. The only question that remains then is, how is this molecule protecting against diabetic heart disease in the first place? And knowing what we now know about the diabetic heart's preference for this pollution-prone fuel in the form of fatty acids, I thought that this microRNA may play a role in preventing that preference, or at least refining it. And sure enough, fatty acid deposition was decreased by higher levels of this microRNA, which was able to do so through increasing the heart's ability to consume fatty acids by fine-tuning the heart's metabolic machinery, the factories that produce the energy from the fuel source, the mitochondria. On a grander scale, this would be equivalent to burning as much dirty fuel as we would like, whether it be wood, coal, or gasoline, without polluting our environment at all, solving both our energy and environmental crises in a single answer. Sadly, this microRNA is limited to affecting just our heart's environment. We hope that these discoveries will give diabetic patients a therapeutic option in the form of this microRNA in order to decrease the risk of developing diabetic heart disease, thus giving them a better chance of living normal lives. Thank you very much for listening.
Great. Well, thank you so much, Tyler. It sounds like a very interesting project, and I'm wondering if you wanted to share with our audience what inspired your work on this.
Sure. So I'm a big believer in drawing inspiration from my personal experiences and the experiences of those around me for my work and pretty much everything else. And I didn't necessarily know what I wanted to do research with until I got halfway through my undergraduate career and my little brother developed type 2 diabetes. And so then I think it was pretty clear that the only answer was diabetic heart or just diabetic research. And I'm really sort of comforted by the number of panelists here. I think there's 3, maybe 4, including me, that are doing diabetic heart research. So, so good on them.
Awesome. Well, I think that's well said. And I think, as we often say at Research America, we like to put patients at the center of all that we do, and we're all patients. So thank you for your research. For our final speaker today, we have Lindsay Olita. She is a protein chemist at NextCure. Her flash talk is titled Determination of Endotoxin in Drug Product and the Impact on Patient Safety. Lindsay, whenever you're ready.
Hello, everyone, and thanks for tuning in. When a sick patient receives their medication, the last thing to be worried about is their own medication, making them more ill from the presence of bacterial endotoxin in the drug. So what is endotoxin, one might ask? Endotoxin is a substance that's on the surface of bacteria, which the arrow is pointing to in the graphic on the right. When endotoxin is detected by the immune system, it triggers a pyrogenic response, which is a fever, and sometimes septic shock, which is when the immune system has an overreaction to endotoxin in the body and ends up damaging cells and eventually vital organs. This kind of immune response can be deadly. It is critical that quality control personnel ensure that there is not a dangerous amount of endotoxin in the drugs. For a long time, there was not an easy way to test for the presence of endotoxin in drug samples. The drug product would be delivered to an animal model, typically a rabbit, and if an immune response was observed, that would indicate the presence of endotoxin in the. Luckily, today, there is an easier way to test for endotoxin. In 1956, it was discovered that elements within horseshoe crab blood have high affinity for binding with endotoxins and will form a clot. Quality control personnel can purchase these cartridges that contain these elements of horseshoe crab blood that will bind to any endotoxin present in the drug samples and form a clot, which can be detected by a ray of light and using a machine. Based on the results of the test, quality control analysts can determine if the drug will be safe for patients to use, based on regulatory guidelines. It's a quick and simple test, but nonetheless, a very important one to make sure that patients receive the high quality drugs that they need without the fear of endotoxin contamination. Thank you.
Great, thank you so much, Lindsay. And thanks for working so hard to ensure patient safety and making that a priority. I know you are about what you're interested outside of science, so anything fun you want to share with the audience? And before you do, I'll just now invite our judges to head to the deliberation room using the link that I sent previously. And we'll also begin the audience Q&A after this, so feel free to leave your questions in the Q&A box. But I also just want to turn back over to Lindsay and give you an opportunity to talk about your hobbies outside of science.
Oh, cool. Thank you for asking. I am a newly retired Division I athlete, so I'm looking for any hobbies, but recently I'm picking up golf, learning how to golf, and I just started to learn how to garden. So those are my 2 new hobbies. Lots of time in the lab, but not being in school leaves you with a lot more free time than I had in college.
Absolutely. Well, I think we can all relate to that. What sport did you play?
I was a rower.
Oh, wow.
Yeah. I'm glad to be retired. I'll probably never do it again.
Awesome. Well, I think at this point we'll bring back all of our participants and we will go ahead and start our question and answer portion of this topic, which we put questions down there. So if you do have a question for our participants, feel free to drop them in the Q&A. We're going to go ahead and get started with one that we have right now, which is, and this will just be volunteer, so you can just raise your hand if you want to answer this question. But the first one is, would anyone like to share the moment they knew they wanted to become a scientist? Yeah, Catherine, thank you.
I don't know if there was a specific moment, but the reason I wanted to become a scientist was from watching childhood TV shows like The Magic School Bus and Bill Nye the Science Guy. And I was always kind of interested in like figuring out how stuff worked. So that's the reason why I got into science. And it's been so rewarding because with what I'm doing now, I think there's definitely like a potential to have real impact on patients. So, yeah.
Thanks for sharing. Tyler.
I think when I first found out I was really interested in science was sort of similar to Catherine's answer. I was watching National Geographic, and there were two shows that I would always watch. I'd always watch Shark Week, and I'd always watch some show about volcanologists going into volcanoes or something. I wanted to do both of those things. My mom said those were far too dangerous, and so then I sort of settled on something maybe like a little less risky. But yeah.
Please, Brian.
I have a very fond memory in second grade of when your parent would come in to talk about their job. I knew that my dad worked in the laundry room and he helped out with a lot of clients in their work. But rather than talking about his job, he actually did a very effective science communication demonstration where he had each of us blow into a beaker with a dye that would change color when we added our carbon dioxide that we expelled. And so it was really one of my first science communication demonstrations, and he was really the first science communicator that I knew, even if I didn't have the words for it at the time.
It's a great story, thank you. Anybody else, or we'll move on to another question, which is, what has motivated you to work on the project that you work on? Does anybody want to take that one? Yeah, Amelia.
Well, I think I kind of touched on this, that obviously like drug addiction is a huge public health issue. And I think that specifically psychostimulant and cocaine addiction is a really interesting area, since most of the focus is on, and rightly, on the opioid epidemic. All of these overdoses are just causing a terrible human life toll. But actually it's surprising, and not a lot of people know that it's almost up to a third, I think, of opioid overdoses actually include other drugs, including a vast majority of those are psychostimulants and cocaine, and specifically methamphetamines also. And methamphetamines are actually hugely on the rise. In the past couple of years, we've seen at least a slowing down of the opioid epidemic, and that's come alongside a dramatic increase in methamphetamine use. So I think that that's something that public health-wise maybe isn't quite as widely acknowledged. So I think that's an important area.
Thanks, Amelia. And you actually have a question, so we'll do this first, and then we'll go back to this, give folks a minute to formulate their answers, which is, what might be the impact of having too much of your opioid administered? Sorry, can't remember. Kappa opioid.
Yeah.
So what might be the impact of too much?
That's a really interesting, that's a really good point. So and it's confusing because there's many different types of opioids. When you think of opioids, you obviously think of things like heroin and morphine that cause overdoses, they cause respiratory depression, but these kappa opioids don't cause that kind of respiratory depression. So that's not a concern in terms of overdosing at all. Too much of it would just make people feel really horrible. And so there's obviously a natural break on how much a person, they're not really like an abuse liability necessarily. And right now there's an issue with, they do have side effects of making people sleepy. They've actually caused hallucinations in some people. And so that's why we're really trying to develop kappa opioids that have a different pharmacological profile that maybe don't cause these side effects. And that's what a lot of the more recent work is on.
Awesome, thank you. And we'll turn it back to our question about motivation for your project. And I know Jordan, you had your hand raised, so let's go back to you, and you can talk about what motivated you to work on this particular project.
Yes, so in 2015, I did a research experience for an undergrad at Notre Dame, and I joined the lab that I did at the time. And it was my first exposure to what metastasis is and kind of just figuring out the different parts to it. And what I took away, one of the things I took away from that summer was that this is such a complex problem that it's going to require ideas and innovation from totally different areas of science to really address this issue. So I think what motivates me from that is just having that possibility of contributing even just a piece to the puzzle, to this grander picture that we're all trying to figure out, which is, you know, like, how do these cells operate and what are their vulnerabilities? So I think that's like my drive, and the knowledge of it and the ability to contribute are the two factors that drive me the most in this project.
Yeah, I would say that's probably a common answer for everyone, right? It's kind of that discovery and that thing, that motivation of knowing something, that you're the first person to figure this out, can be pretty exhilarating. So we have another question. Oh, this is a good one. What role has mentors played in your scientific development, and what has been the most meaningful advice that you've been given? So if anybody wants to take that, just give a hand raise. And yeah, Jamal.
I would say, like, I think mentors have had a huge impact in my scientific development, and I like to think that most people here would probably say the same thing. And it's because, like, mentors there's different styles of mentoring, and as you like go through your scientific career, you're going to meet people with different styles of mentoring and see what works for you. And especially during the PhD time, I've been lucky enough to like find my mentor, Dr. Elemento, who has like helped me through like rough times, helped me through good times too, but mostly through the rough times, where it's like convincing me to like keep going. And I think one of the most meaningful parts of advice that he's given me is that it doesn't always work out, and that's okay. Which is something, you know, your parents say to you, but like, you know, you grow up and you stop listening to your parents. So like when scientific mentors tend to say sort of things like that over and over again, it's something you can really relate to.
I think failure is such a big part of science, right? It's kind of knowing all the things that you try that don't work, that kind of lead you on the right path. And so that's why it takes years to solve a very simple question, right? Medina, did you want to chime in as well?
Yeah, so what I wanted to say actually goes right along those lines that you just mentioned. One of the best advices that I've received throughout my graduate journey was knowing how to distance and differentiate yourself as an individual from your project. So if your project is not successful, if you don't have successful results, that doesn't mirror onto you as an individual, as a persona. And I think it's very important for our mental health to keep that in mind and be aware of it the whole time.
Yeah, I could not agree more. I think that's a really great point. Thank you for bringing that up. We have a question for Catherine. Is there any evidence suggesting the formulation that you're developing or that you're delivering might have an impact on the efficacy of the polymer?
I think that question may be referring to the efficacy of the drug itself. But so when you just add the polymer as an additive without attaching it to the protein, there's no impact on the activity of insulin. It's exactly the same. When you do attach any sort of polymer to a protein, which is used in the clinic, some polymers have been used as what we call conjugates, where you attach the polymer, that can impact the activity of the drug, because you're adding this large molecule to the protein, and sometimes that can affect binding to a target or binding with some molecule. So this is a concern, but you can alter where you attach it on the protein, which can minimize this effect. So, yeah.
Great, thank you. And we have a couple of questions, unless anybody else wanted to share anything else about advice from mentors. We will move on to the next question, which is, do you find young people are less excited about science? What can we do to ensure young people understand the importance of research and science? Yeah, Amelia.
I hope not. I don't think the young people are necessarily less excited about science than they should be. I think a lot of it has to do with things like accessibility. And, I think that this topic has been touched on a lot in a lot of the really great discussions we've heard the past couple of days about like almost the politicization of science and the idea that it's this like ivory tower thing that only some people have access to or only some people can do. And so I think that there are a lot of really amazing STEM programs for kids and things just showing that like science is fun and exciting and accessible and like you're doing it every day and it shouldn't be, separate from the rest of society and the rest of the world, that it should be integrated into everything we do. So I think that will play a big role in getting kids excited about science.
Yeah, absolutely. And I'll let Jordan also comment on this. But going along with that, we got another question about how can we get students excited about science? So maybe along those same lines. And Jordan, you can comment on the previous question too.
Right. I think my response will piggyback off of Amelia's response. In addition, I would say that confidence in young kids, I think that math and science are still very daunting subjects that people tend to shy away from. So just having these programs and having the mentors, which in that question about mentors plays a role here, showing kids that you can do this. You know what I mean? I think just being able to see yourself in these positions or in these lab spaces, in these industries, actually doing the work yourself, then creates that accessibility piece that Amelia was talking about. It also builds that confidence that, oh, I can do this. And I think that would definitely increase the presence of young people in science. It's just knocking down that intimidation factor in the fields.
And Tyler, do you want to also make a comment?
Definitely, yeah. So for a couple of years, I helped put on a conference centered around getting middle school aged girls interested in science. And what that would be, we would just host it at the sort of a university, and we would have a series of workshops. And I found, that the easiest way to get young people, especially very young people, interested in science is just make it engageable. Make it interesting. Don't even mention science. It seems maybe counterintuitive, but just show them what's really, really cool. And to also piggyback off of what Amelia said, I currently work with a nonprofit here in Omaha that's centered around prescription drug abuse in the youth. And so my role in that nonprofit is to organize workshops to educate the youth, if you will, on the sort of long-term side effects of drug addiction, prescription drug addiction to their brains. And so that's sort of the other side of the coin where in that case, you'd like to mention science because it's actually directly impacting their lives. And so whichever way you do it, as long as you engage, I think that's the most important bit.
Thank you. And Medina, you also wanted to make a comment?
Yeah, I wanted to add more onto the part of the question, how can we engage young people and how can we make them more excited about science? I think the biggest fear is, as a human, in my personal opinion, the biggest fear that I could have is the fear of the unknown. And when people don't know the both sides, I think they tend to stay away from it. So, we all are confident and, comfortable sharing our success stories. But as current scientists, I think it could be useful for, younger scientists to share and show them the sides of our research that are not so successful, to just show them everything has those two sides and it's okay to fail and to make mistakes, you just learn and become better throughout those experiences.
Yeah, I think that's a great point. And I know I've heard from some very prominent people, they actually make failure resumes for people to show everyone that even people who are successful have actually failed at a lot of things, and that's okay, and it helps you learn and grow. So appreciate your feedback. We'll go to our next question, which is, how do we make science more accessible to a non-scientist audience? Does anybody want to take that one? Yeah, Jamal.
I think there are many approaches, and I think that all of us today did a great job of explaining our research in that way. I'm going to piggyback off of what Amelia said, though. There is this mindset that you say scientific jargon, we're in this ivory tower. Oh, I'd never understand that. And it's kind of like, no, it's actually, you just need to like, I would say it's kind of breaking it down into what Tyler was saying and like being engaging. So it's really exciting was like, especially like coming up with talks like these, it was kind of like figuring out what is the analogy that you're going to take in order to make science more accessible. And if that analogy is not good enough, like what other steps? And it's kind of like, it's okay to like slowly ease people into that. It doesn't have to be automatically know exactly what a polymer is or automatically know what this single protein does, because scientists today are not always experts in completely everything. So I think there's multiple ways, and it's really exciting to see that everybody has been doing it, at least in this environment.
Absolutely, Jamal, and I'm so glad you brought that up, because I do really think it's events like these and trying to, you know, get the word out there about what you're doing in a way that, you know, and really is an engaging way to draw people in, and not necessarily in a sense making it, you know, less complicated, but really just putting in a way that, you know, anybody who wants to who displays an interest in what you're doing can really like find that hook and get really interested, and you can inspire more people to work on science. So I think that's really exciting. We have another question, which is, have you, or obviously you have, what, maybe can you talk about a challenge you've overcome, or a particular stereotype in pursuit of science? Has anybody experienced any challenge that they'd want to share about their research or working as a scientist? Yeah, Lindsay.
I think I can speak to this one. I'm by far the youngest person at our company, having only been out of undergrad for like a year. And a lot of my peers are like in their 30s, 40s, like have at least like 10 years of experience. And so one of the stereotypes that I caught on to really quick was, oh, you're young, like you don't really know what you're doing, or young employees tend to not work so hard. You know, they're out the door, you know, at 5 or whatever, which is not something that like, you know, I stood for, like I believe in. So you kind of have to, when somebody tells you what to do or is explaining things to you, just like, you know, take it and be grateful for their even explaining anything to you, and then, you know, just do what you got to do.
Well, I think that's great advice. And I think especially thinking about, you know, trying to inspire the next generation and getting everybody excited about what you're doing. It might be disheartening to hear feedback from older, you know, mentors or people you might consider looking up to, and them telling you that you're not, you know, or that they have these preconceived notions. So I think that's really an important point. We have question, and I'm not sure if anybody, if you don't want to comment on it, you don't have to, but how can the US be more science friendly? Does anybody have international experience or thinking about science in the broader context? Maybe perhaps if you are, you know, maybe you know people who are part of the international science community and they are experiencing some challenges at the moment, or anybody want to comment on that? Yeah, Brian.
Yeah, it's a good question. I think I can share an anecdote from a friend who got her PhD in Sweden. And in Sweden, they require a plain language summary of the thesis, which I think would be very helpful for all dissertations, and then as well for preprints that have really seen a rise in use during COVID. Having a plain language summary of preprint articles, so that when they are picked up by news outlets and distributed, there's maybe a greater understanding and less is lost in translation.
Yeah, I think that's a great point. Thank you for sharing that. Our next question is, do you see yourself as an advocate for science? So are you comfortable talking about policies around it, or are there things that you want to share about how that maybe translates, how your research translates into policy? Yeah, Tyler.
This is a really interesting question for me, because something I do on the side is I'll take trips with a couple of other students down to the state capital of Nebraska, down at Lincoln, And we'll talk with the state legislators about bills that our medical centers sort of deemed priority. And when you talk to a legislator about science, it's just like talking to an average person about science. But when you're at sort of the stage of your career, you've got the advantage of being a student, essentially. And so I found that they're very receptive to that kind of an idea. So if you are thinking about advocating for policy, I think right now is the best time to do it in your career, because you're learning everything surrounding science, but you've also got the advantage of when you go to a state representative or any sort of representative of being a novice and being more genuine and increasing the reception of the ideas you're trying to put out. And so through that work, we were able to get a, I think it was something like a $5 million allotted amount from the state funds out of committee to go to the floor to be voted on the state Senate. And so if you aren't advocating, it's actually really awesome and very easy. I highly recommend you find a group to do it.
Thank you. Does anybody else want to share? Yeah, Amelia.
Yeah, I think maybe it was one of yesterday's panels. It was Dr. McMurray Heath was talking about kind of some things that went wrong and that led to the opioid epidemic in the way it is now. And one of the things she was saying was that scientists did tend to, I think she said, like, stay in their sandbox instead of going out to the bigger playing field, which I thought was really, really good way of putting it. And that, you know, we have a good understanding of the underlying science of things like addiction and how to treat it, but that it maybe goes against some intuitive thinking about the issues, or, you know, the preconceived notions and biases people have. And so I think that, like, scientists being able to advocate in that space would be incredibly beneficial. But I think that some more maybe formal training and opportunities for scientists to get involved would be great. I mean, I think it's fantastic that Tyler and, you know, all of those scientists involved in Research America and all of these different organizations have that opportunity. And I think having that more widely accessible could only help. I think Caitlin. Okay, there you are. Caitlin, I think you're frozen. Oh, now she's gone.
Okay, am I back? Hello? Okay.
Yes.
All right, so I'm told our judges are still deliberating, which is just a testament to how tough the competition was. But I also just wanted to go ahead, and I know Jordan has shared, I'll go ahead and turn off my video because we have a storm here in DC and I think it's affecting my internet connection. So anyway, Jordan's already shared her plans, and I know Catherine and Lindsay are both working hard at companies, but if anybody else wants to share their future plans or how they want to integrate policy or science communication into their future plans, I'm sure the audience would love to hear about that as well. Yeah, maybe Brian, if you want to kick things off.
Yeah, happy to share, considering I'm also supposed to be writing parts of my dissertation right now and putting out a paper as well. But in addition to working on my research, I've, for the past couple of years, worked with a policy and advocacy student-run group here at Rutgers, and have really thought about how can we help Rutgers live up to its mission statement of serving the people of New Jersey and really increasing the trust and building relationships between Rutgers scientists and the public. And so that's something I really would like to continue beyond after my defense. And so I'm applying for several of the policy fellowships that have deadlines upcoming in the next couple of months. But in between, I haven't really completely planned out what I'm going to do until those policy fellowships would start sort of next year. But I am sort of excited to have some time to kind of decompress after finishing up my PhD.
Absolutely. I think we can all relate to that. I also wanted to ask about, obviously COVID-19 has impacted research labs. Does anybody want to share sort of an on-the-ground experience of how COVID-19 has affected maybe your research timeline, or even, you know, I know some people are working in shifts and there's all kinds of these protocols that have been in place. So are there any things that you want to share about your experience being a researcher during COVID-19? Yeah, Medina, please.
I can start with this one because I guess my what I'm going to share is positive, so I can keep it on positive notes so far. So being a computational biologist, I was one of the lucky ones who has not been influenced, whose work has not been influenced that much, you know, with the whole pandemic going on. So as long as I have, you know, computer, decent internet connection, I am good to do my work.
Great. And Jordan?
So I did not have the fortunate opportunity of being a computational biologist. I am a wet lab cell biologist, and it was interesting because I did my candidacy exam, I think about a week or so before the lockdown and the shut-in started happening. So I was able to hop on Zoom and meet with my committee members, do my candidacy, obtain candidacy, and then after that, we took 3 months off of no lab work. It was essentially just looking up papers, putting together whatever figures we could. Some people were able to put together review papers, just finding other ways to just keep ourselves productive and progressing towards our degree was the objective during those 3 months. And then around June 8th is when the lab started opening up in shifts. And I think we progressed through the phases pretty nicely. And since then, I think things have been running smoothly for grad students in the labs, and those have been good. I think a positive note out of all that, though, is that we have 8 a.m. lab meetings, and instead of coming in physically into the lab, we just stay home, whether it be in your bed or in your office, and you attend meetings from your computer, which I personally am an advocate of this. I think that should continue post-COVID vaccine.
Absolutely, thank you. And I see our judges have returned. So I'm sure that was a tough deliberation. I wanted to go ahead and invite them to say a few remarks about the competition today, starting with Joel.
Sure, thank you, Caitlin, and really amazing job, guys. Just a little introduction for myself. You may have seen some background. So I work in the pharmaceutical industry and had made a transition out of academic research into the pharmaceutical industry. I've worked with Research America a long time, but this is my first judging effort for a program like this. Caitlin and the Research America gang did a really good job, but I have to commend all of you to get all of that information packed into 3 minutes. I think each of you had a lot of really great information to share. And I can't thank you enough for the work that you put in it, but most importantly, the work that has gone on around these 3 minutes. So congratulations to everybody.
And next up is Jocelyn.
Hi, thank you. I just want to echo what Joel has said. You are all winners. The competition was fierce, which is why our deliberations took a little bit longer, but each presentation was fantastic. The research that you all are doing into finding cutting edge solutions to some of our toughest challenges is just awe-inspiring. And one particular area, well, several particular areas, not only in cancer research, but also the opioid epidemic, all of these areas are critical to our long-term success as people and as a country. So this is my first time a competition like this. I feel like it's my American Idol moment. And so super excited to be a part of this with all of you. And as I said, you are all winners. This has been truly fantastic. So hats off to you.
Oh, American Idol, that's a tough one to follow. Willie?
Sure. So I'd like to echo my colleagues and congratulate you all on what a wonderful job you did. So I'll share, I'm Cuban, and there's a joke within Cubans that Cubans are never left speechless. And so I was left speechless, which really does say quite a bit. And I feel that all of us really have to be incredibly optimistic about the future of biomedical research because of the great work that you're doing. So keep up the great work, and I think you made us all very, very proud.
Thank you. And Rush, Dr. Holt.
Thanks. I will echo what all the others have said and comment that I was particularly impressed that all of you made it clear that this is science not for scientists, but science for people, in this case, patients. I think that's so important. And, you know, I think in this age of misinformation and disinformation, the job for every scientist is not just to explain his or her research clearly and in detail, but also to every audience all the time, show the process, show why science works. How using evidence from which you've tried to remove bias is used to displace preconceptions and opinions and hunches. That is a lesson that everybody in America, really around the world, needs to know, even more than they need to know the details of your research.
All right, thank you so much. And I'd like to invite Jenny LeRae to come on and talk about the introduction for the winners as that time has come. So I'm going to go off camera to try to save my internet connection. And Jenny, if you're on. Okay, I'm receiving where Jenny is not on. Well, I think the judges have all deliberated and understand the rankings with contestants. So I am also anxious to hear how that's going. But maybe we can start. I think Rush, Dr. Holt's going to introduce the third place prize.
Yeah, in third place, and of course it could have been second place, or it could have been first place, or it could have been any of you. But in third place, receiving a cash prize of $500, congratulations to Jordan Cockfield. It was a wonderful talk with all the ingredients, really compelling.
Thank you so much.
Congratulations, Jordan. Awesome job. Okay, Jocelyn is going to announce the second place prize.
Absolutely. So congratulations, Jordan. Drum roll, please, for our second place winners. We actually have a tie, and I will announce winners in alphabetical order. So our first second place winner is Tyler Cambus. Congratulations to you, Tyler. You will receive a cash prize of $1,000. Excellent presentation. We loved it. And so you are our first winner. Our second prize winner, another drum roll please, is Catherine Messina. Catherine, congratulations to you. You will also receive a $1,000 cash prize. I mean, both of your presentations just absolutely amazing, and we are super excited that we get to recognize you both in this way. So congratulations.
Thank you.
Congratulations. And Joel, to announce the first place prize.
Yeah, well, thank you, Caitlin. Not only do I get to go first in making my comments, I get the best position to announce first place winner too. This was a hard decision, and I'll just congratulate all of you once again. Congratulations to the third and second place winners. But I'd like to announce the first place winner receiving a cash prize of $1,800. Congratulations, Amelia Dunn, for her talk, and just really fantastic. I was looking at my notes, Amelia. I think this has, you know, incredible application to life now and life in the future too. So incredibly focused, patient-focused, society-focused research. So congratulations, very well done.
Thank you so much. Really appreciate it. Everybody did such a good job.
Oh, yes, it did. Congratulations to all of our competitors on a terrific display of science communication, and congratulations to our winners. All of our contestants are receiving a cash prize of $200 for doing such a great job today. A special thanks to our judges who obviously had a very difficult task today. And our judges and contestants can now leave and join me in the reception room in a few moments. I just wanted to take this time for the audience to thank you for your participation in joining us today. You can go ahead and head back to the auditorium for our next panel. Thank you.